Hi everyone. In this video, we're going to look at the cell cycle and some of the checkpoints that help to regulate it. The cell cycle can be thought of as the life of a cell, from the moment it's made to the moment it splits into two. The actual division part of the cycle only accounts for around 10% of the cell's time, with the rest of it being spent in interphase, where the cell is performing its regular functions and preparing itself for division. We can break interphase down into three main subphases, the first of which is referred to as G1, or the first gap phase. During G1, a cell is growing, replicating its contents from proteins to membranes and even whole organelles. In fact, the only cellular component that isn't replicated during this phase are the chromosomes. After G1, the cell will move into S phase, where the DNA will be replicated. The chromosomes wouldn't actually be condensed enough to be visible at this point, but each one will be copied and attached to its duplicate, which we refer to as sister chromatids. These are attached the whole way along the chromosome arms by proteins called cohesins, but are most tightly attached at a region called the centromere. Once the chromosomes have been replicated, the cell can progress through G2, the second gap phase. During this time, the cell is mostly checking for any errors to ensure that the daughter cells produced by division will be accurate and preparing for the process of division, which will happen during M, or the mitotic phase, which consists of mitosis and cytokinesis. We'll look at these processes in more detail in another video, but they result in the division of both the chromosomes and the cellular content, splitting one parent cell into two daughter cells, and bringing us back to the beginning of the cycle. Like everything else in biology, the cell cycle has to be controlled in order for things to function correctly. In fact, one of the hallmarks of cancer cells compared to normal cells is the lack of cell cycle control, leading to rapid cell division. As the cell progresses through the cell cycle, it will encounter several checkpoints, the first of which occurs towards the end of G1 phase and is referred to as the G1S checkpoint. This serves as a stop signal, forcing the cell to pause the cycle momentarily so that checks can be carried out. At the G1S checkpoint, cell contents are checked to make sure that the proteins necessary for DNA replication have been produced, and to ensure that the cell has sufficient nutrients, other contents, and mass to support dividing into two. Now, this is typically the most important checkpoint for cells. If they make it through the G1S checkpoint, they'll likely make it through the others to division. If anything is missing or insufficient, the cell will enter a resting phase called G0 to give it time to fix the issues. If everything is fixed, then the cell can go back into the cycle to continue into S phase. The next checkpoint comes near the end of G2 phase, where the cell will check for errors that occurred during DNA replication or any DNA damage. Again, if anything is found to be incorrect, the cell will activate DNA repair machinery to try to fix the problem. If there are errors that can't be addressed, the cell will go into a program of cell suicide called apoptosis to prevent errors being passed down to the daughter cells. Assuming everything is fixed, however, the cell will continue into M phase, where it will encounter a final checkpoint termed the M or spindle checkpoint. At this point, the cell will check to make sure that all of the chromosomes are correctly attached to the microtubules of the mitotic spindle before separating them. If any unattached chromosomes are found, the cell will attempt to pick them up and attach them. Once that is completed, the cell can continue through the rest of mitosis and cytokinesis, forming its two daughter cells. In addition to the negative control of checkpoints, there are also additional positive signals regulating cell division. Cyclins, for example, are proteins whose concentration fluctuates throughout the cell cycle, typically reaching their highest level at one of the checkpoints that we just discussed, to stimulate the progression from one phase to the next. As the cell moves through G1, the concentration of G1S cyclins will increase, as shown by the blue line on the chart here. The G1S cyclins bind to a type of protein called a cyclin-dependent kinase, or CDK, to stimulate its activity in phosphorylating its targets, as shown by the yellow curve on the chart. And this will help to push the cell from G1 into S phase by stimulating DNA replication. Similarly, S cyclins will increase in concentration as the cell heads through S towards G2, activating their CDKs. M cyclins will continue to increase until the G2M transition and form a complex known as maturation promoting factor, or MPF, which is required to start the process of mitosis. MPF also activates a protein complex to cause its own breakdown, allowing the cell to progress out of mitosis and back into G1. At each checkpoint, CDK inhibitors act on the cyclin CDK complexes to slow down progression through the checkpoints, giving the cell time to repair any damage or fix any other errors that need to be addressed before moving on to the next stage. Thanks for watching, and please be sure to check out the video showing the processes of mitosis and cytokinesis.